Hi, this lecture is about primary production. This is being recorded for my Florida Gulf Coast University Marine Ecology class, but anybody who's interested in primary production, particularly primary production in the marine environments, might find it interesting. So, what is production? Production is a process of making stuff. What is produced when we're talking about primary production? In primary production, the stuff that is made is organic matter, the material of life, and it is made from inorganic matter, small simple molecules, and it is made by organisms that we call primary producers. More detail on this will be on the next slide. So organic matter is the stuff that is made in the process of primary production, and organic matter is a set of an infinite variety of complex molecules that all share a carbon framework. So these complex molecules that we call organic matter includes classes of molecules like carbohydrates, lipids, which are fats, proteins, nucleic acids like DNA and RNA, and any of the complex large molecules that make up living things. They're all what we call organic matter. So carbon is obviously the main framework of these molecules, but they also include many other types of atoms like hydrogen, oxygen, nitrogen, phosphorus, and sulfur, and other elements as well can be found in some organic molecules. So these large complex molecules don't come out of nowhere. They're stitched together from smaller, simpler molecules, which we call inorganic matter. So some examples of the small, simple molecules that are assembled into larger, more complex molecules include carbon dioxide, water, ammonia, phosphate, and other simple molecules that are stitched together by these organisms called primary producers into the large, complex molecules of life. The process of stitching these small molecules together, if it uses light as the energy source to accomplish this, it's called photosynthesis. And if it uses chemical energy to fuel the synthesis, it's called chemosynthesis. The reason we care about primary production is because it's the ultimate source of all the organic matter on Earth, including the organic matter that comprises our own human bodies. So the primary producers are able to assemble this organic matter from the raw inorganic materials, the tiny inorganic molecules, whereas organisms that are higher up the food chain, like humans, we don't make organic matter from scratch. We get our organic matter from the things that we eat, and the organic molecules are therefore transferred up the food chain from the base of the food chain, which is the primary producers, also known as autotrophs, up to the consumers, also known as the heterotrophs. So that's one of the reasons that we care about primary production. The other reason that we care about primary production is because one of the byproducts of primary production, byproduct of photosynthetic primary production, is oxygen. So as organic matter is being produced by these plants, they're also making oxygen. And oxygen is super important for all higher life forms which use oxygen in their metabolic processes. We require oxygen to break down our food and to use energy. So we need oxygen as well. So we need the living material that's made by the primary producers, and we also need the oxygen that's made by the primary producers. So in ecology, there's this term called bottom-up control, and it refers to the bottom of the food chain. The bottom of the food chain is the primary producers, the plants in a photosynthesis-based food chain. So the idea of bottom-up control means that what's happening higher up in the food chain is totally dependent on what's happening at the bottom of the food chain. So here's two example food chains, the one on the left from a terrestrial environment and the one on the right from a marine environment. Both of these food chains begin with primary producers, large multicellular plant in the terrestrial environment and microscopic algae in the marine environment, but in both cases the food chain works its way up from these primary producers to ultimately to top predators. And how many hawks you can have or how many sharks you can have in the ecosystem ultimately depends on the primary productivity at the base of the food chain. So really everything depends on the primary producers. We're not going to go into great depth about the cellular and molecular mechanisms of primary production, but there are some things that I would like to remind you of. If you've taken a general biology course, you probably learned in more detail the cycles and chemical reactions that are occurring during the process of photosynthesis, but I'm just going to cover the very basics here. And the very basics are that the two raw ingredients that are being combined to form the basic type of organic matter in primary production are carbon dioxide and water. And carbon dioxide and water are stitched together with the power of sunlight to create organic molecules with this approximate ratio of carbon, hydrogen, and oxygen. And there's also a byproduct created, which is oxygen gas. So these two products of primary production are organic matter and oxygen. This is going to come into play later because there's a proportionality between the amount of organic matter produced and the amount of oxygen produced. So sometimes if you want to measure primary production, you actually just measure the amount of oxygen that was produced, and you can relate that to the amount of organic matter that was produced. That's why it's important to know the chemical equations for this. In chemosynthesis, this type of primary production that does not use light as the energy source, but instead uses energy-rich compounds like hydrogen sulfide as the energy source. Some of the chemical ingredients are a little bit different, but carbon dioxide is still one of the raw ingredients, and organic matter, of course, is still the product because primary production is the production of organic matter. Photosynthesis happens in sunlit parts of the earth like the surface waters of the ocean and the terrestrial environment. These organisms here are phytoplankton, microscopic single-celled plant-like organisms that live in the ocean. And in the picture for chemosynthesis, we're looking at a deep-sea hydrothermal vent environment where energy-rich chemicals burble out of the seafloor, and chemosynthetic bacteria harness those energy-rich chemicals to do their production of organic matter, and the other organisms in that food chain eat those chemosynthetic microorganisms. So the common theme for these two sort of different types of primary production, the light-based one and the purely chemical one, is that an energy source is harnessed in order to synthesize complex molecules from simple ones. So you're taking simple molecules and an energy source and doing these chemical reactions to turn them into complex molecules that we call organic matter. So throughout this lecture, when I'm talking about photosynthesis, I'll use the word plants to refer to any photosynthetic organisms. And this is a little bit informal because really, technically speaking, plant means just a certain class of complex photosynthetic organisms that have true roots, leaves, seeds, and vascular tissues. And there are some true plants in the oceans, in shallow waters and along the edges of the ocean, but most of the plant-like organisms in the ocean are actually evolutionarily more primitive. They include prokaryotic organisms like cyanobacteria and relatively simple eukaryotic organisms like single-celled eukaryotic algae and multicellular eukaryotic algae that we call seaweeds, such as the red algae rhodophyta, green algae chlorophyta, and brown algae phaeophyta. So lots of organisms that are sort of in different locations on the evolutionary tree, but which all do photosynthesis, and for the purposes of presenting the information, I'm just going to refer to them all as plants, even though that's not technically true. So plants have been around since not too long after the very first life formed. The very first life that arose on Earth through the process of abiogenesis was non-photosynthetic. It was chemosynthetic. So chemical energy was the original energy source for the production of organic matter by the very first organisms in Earth's history. But as early as 3.5 billion years ago, we think that photosynthesis had evolved. The first forms of photosynthesis were more primitive than the modern kind, less efficient. They did not produce oxygen as a byproduct of that early photosynthesis. And oxygenic photosynthesis, the type that's most common today, arose about 2.5 billion years ago. And we think that ancestors of today's cyanobacteria were the first organisms to do oxygenic photosynthesis. When that type of photosynthesis evolved, it really changed the atmosphere of the Earth as the oxygen that was produced transformed the atmosphere and actually made it really difficult for some existing types of life to survive. But eventually life did adapt, and now many types of advanced life use the oxygen, and so that was a big change early in Earth's history. About 1.6 billion years ago, the single-celled algae sort of had an evolutionary leap where they went from being those simple prokaryotic forms to being more complex eukaryotic algae. We think that one of the ways that the eukaryotic algae evolved was through endosymbiosis. So there were these simple prokaryotic algae that got engulfed by eukaryotic cells, and then instead of being digested, they actually developed a symbiotic relationship where they lived inside the cells that had sort of engulfed them, and then eventually they fused into one organism. And this happened, this endosymbiosis kind of thing happened multiple times in the evolution of algae, and makes the evolutionary tree of the plants and algae pretty complicated, because it's not like the branches are just separating. There's some of the branches that were coming back together through endosymbiosis. Anyway, by 600 million years ago, which was around the time that the first complex multicellular animals were developing, we were also getting multicellular algae, so complex seaweeds were leaving their fossils behind by 600 million years ago. And by 475 million years ago, some of those seaweeds from the green seaweeds group were developing features that allowed them to survive being out of the water for a little bit. So plants began to colonize the land, and they evolved from this particular type of algae called the green algae. And plants over the millions of years developed more adaptations to live and reproduce on land, seeds and pollen, roots and complex veins and leaves that allowed them to expand on the land. And one of the features that actually didn't evolve until relatively recently in land plants was flowers. But flowers were really a great adaptation that helped plants evolve and adapt faster, reproduce efficiently. And some of the plants that have recolonized the ocean, like the seagrasses and mangroves, are actually descended from land-based flowering plants. So whether plants are living in the ocean or on land, they all need some of the same basic ingredients to survive. They all need sunlight as their energy source. They all need water and carbon dioxide as the raw inorganic molecules for their photosynthesis, and they need nutrients, chemicals like nitrogen and phosphorus, to form some of the organic molecules that have nitrogen and phosphorus in them. So what differs between the land plants and the ocean plants is where they get those four things. So on land, the nutrients and water tend to be concentrated beneath the soil, and so plants have these root structures that suck up the nutrients and water from the soil, and then the above ground structure of the land plants is very different from the below ground structure, because the above ground structure is adapted to capturing sunlight and carbon dioxide. In the ocean, they're absorbing everything from all around them, so they don't need to have this differentiated two-part body structure. They just absorb the nutrients and carbon dioxide and water from all around them. But one of the limitations for water plants is that only the surface of the layer of the water down to a certain depth has enough light for the plants to grow in. So the plants need some way to stay near the surface, and usually that's by being really small so they don't sink rapidly, because if these plants sank to where there's not enough light, then they would die. So there are many different types of microscopic plants in the ocean, collectively referred to as phytoplankton, and I'm not going to ask you to know them all, but I would like you to know some features of some of the plants in the ocean. So one of the major groups of plants in the ocean is the diatoms. Diatoms are eukaryotic algae. In fact, these first three groups that I'm going to talk about are all eukaryotic organisms. And diatoms have a glass shell, a silica dioxide shell. SiO2 is the molecular formula for glass. They also like high levels of nutrients, and so diatoms tend to be found in areas where there's upwelling or nutrients that are delivered from coastal runoff. So glass shells like high levels of nutrients abundant near shores. Another type of eukaryotic algae that has a different type of shell is the group called coccolithophores. And coccolithophores have a calcium carbonate type of shell, and their shell is sort of comprised of all these intricate calcium carbonate plates that cover the organism. And the coccolithophores tend to live a little further offshore in environments that aren't quite as rich in nutrients, but they can still be really abundant and form an important base of the food chain for the oceans. The next group of eukaryotic phytoplankton that I want you to know about are the dinoflagellates. They have the word flagella in the name, and as the name implies, they have not one but two flagella per cell. So the flagella is like the tail of a sperm cell, and so the dinoflagellates have one that they use for movement and one that they wrap around their waist, and they can use to sort of spiral as they move through the water. I mean, they're single cells. They don't really have a waist, but there's sort of this groove on the center of the cell, and that's where this flagella sort of fits into this groove, and that helps them sort of spin as they move through the water. So you don't usually think of plants as moving, but when we're talking about single-celled plant-like organisms, the lines are a little bit blurry, and many of these photosynthetic organisms actually are mobile. The dinoflagellates, they don't have any type of special shell or skeleton, but they have some other special abilities. Some are bioluminescent, so you can see the ones glowing in this picture. Others are able to produce toxic compounds for defense and other purposes, and that can make them harmful when they're in large numbers because they can form what we call toxic algae blooms or harmful algae blooms, like red tide is a type of harmful algae bloom caused by a type of dinoflagellate. The last group of marine phytoplankton that I want you to know about are the cyanobacteria. So the cyanobacteria include a really high diversity of organisms, from simple little round balls like the Synechococcus algae to more complex clumps of cells that are adhered together in filaments like this trichodesmium. Some of them are, like the Synechococcus, are adapted to living in low light levels where there's just barely enough light to grow. Others, like the trichodesmium, they live in the bright waters right at the surface of the ocean. And one of the sort of special abilities of the dinoflagellates is they can turn nitrogen gas, N2, into more usable forms of nitrogen like ammonia, NH3, and this means that they can find a nitrogen source where there would not otherwise be enough nitrogen for primary production to happen, and so they can sort of make primary production possible in nutrient limited areas. To measure primary production, we need to be careful about the terminology that we use. So primary production overall is the production of organic matter, but there's sort of different terms that describe the gross production versus the net production, and they're related by how they deal with this other thing, respiration. So I'll explain those three things: gross production, respiration, and net primary production on this slide here. So gross primary production is the overall rate that producers create organic matter. So basically, how fast are they taking carbon dioxide and water and smushing them together into complex organic molecules? So the units that gross primary production is typically measured in are grams of carbon per meter squared per year. So to sort of picture that, you can picture a meter squared sized patch of ocean, or maybe for convenience we can just picture a meter squared sized patch of lawn. Most of us have seen, grass grow in a lawn, and so in that 1 meter squared area, the amount of growth in a certain amount of time is the primary production, the gross primary production. So if you measured that over a year, how much grass was made by this patch of ground, you could express that gross primary production in grams of carbon per meter squared per year. And one of the things that complicates the measurement of primary production, though, is that just as fast sometimes as organic matter is being produced, it is being destroyed. And what destroys organic matter is the process called respiration. And all organisms, even the ones that are primary producers and make organic matter, they also have to burn organic matter. So they can sort of store energy as organic matter, but they need to get it back to do the things that keep them alive and growing and reproducing. So they need to burn that organic matter through this process called respiration. So there's some rate at which they're creating organic matter, but there's also some simultaneous rate of respiration where they're destroying organic matter. And so it's really the balance between the gross primary production and the respiration that determines the overall amount of organic matter that's produced in a given time, which is the net primary production. An analogy that I sometimes use is imagine it's a restaurant, and the sales of the day, the total amount of money in the cash register at the end of the day is the gross primary production, but then for the restaurant to maintain itself, it has to buy the food and pay the staff, and so you have to sort of expend that to keep the system going, and that's the respiration. And then sort of what's left over as the overall profit is the net gain, the net primary production. So you've got your gains and your losses and the balance between them, which is the net primary production. So net primary production, it can be positive if you're making gains, but net primary production could actually be negative if you're sort of running a deficit and sort of like a restaurant that lost money that day. It wouldn't be able to sustain that forever because it would eventually go out of business, or if this is a plant, it'll eventually run out of energy reserves and die out. But net primary production can be positive or negative or zero, and that's why it's really important to know what net primary production is, because it sort of tells you, is this production sustaining itself or depleting itself? And these are things that scientists like to know. So the thing about gross primary production is that it's hard to figure it out. You can't really measure it directly because you can't get a handle on something that's being created and destroyed at the same time. So what you have to do is measure the total amount of primary production that you end up with, net primary production, and measure the amount of respiration. And from the difference between those two, you can calculate gross primary production. So here's how that works. We use as our indicator of primary production oxygen. The reason we use oxygen as our indicator of primary production instead of organic matter, which would be a little more intuitive, is that it's easier to measure oxygen than it is to measure the amount of organic matter. So primary production is the amount of organic matter that's produced over a given time, but since it's hard to measure that, we measure the amount of oxygen that's produced over a given time, because that is proportional to the amount of organic matter that's produced over that same time, due to the balance of the equation here. So we put everything in terms of oxygen, and then later on, if we want to, we can convert that into whatever the amount of organic matter was based on the equation. So net primary productivity, NPP, we measure that as the amount of oxygen produced in the light. And respiration, we measure as the amount of oxygen used up in the dark. Respiration happens in the light also, but you measure it in the dark so that you can make sure that you're measuring just respiration and not that mix of respiration and gross primary production, which is net primary productivity. So in practice in the field, if you're a marine biologist and you want to figure out the primary productivity of the ocean, you go out in your boat and you take a sample of water, which includes all of the plankton in the water. You probably collect it in a big bucket like this, and then you can divide that bucket up into smaller jars. Clear jars that let light in, and opaque jars that are dark brown or black and don't let any light in. And you put oxygen probes in the jars. You measure the oxygen level when you start, which should be the same in each jar at the start because they just came straight from the ocean. But then over time, the jar that's in the light will probably have an increasing amount of oxygen because there's photosynthesis happening there, and if it's in the light, the photosynthesis is probably exceeding the rate of respiration, so oxygen will build up, and you'll get this change in oxygen. Triangle means change in, so change in oxygen levels in the light, which are proportional to net primary production, and then change in oxygen levels in the dark, which are proportional to respiration. And then you combine the two. So to combine the two to figure out gross primary production, you take the change in oxygen levels in the light, and then you account for the change in oxygen levels in the dark and add that in, and you figure out the gross primary production from that. So this works if you have a pure sample of just primary producers like just phytoplankton. But what's actually more likely is that in your jar of ocean, you've got not only the microscopic plants, but you've also got microscopic animals, the zooplankton in there as well. So you've got some organisms in there that are not primary producers at all, that are just consumers. And so they add to the respiration without contributing any to the primary production. And so the overall change is a result not just of how much of the photosynthesis respiration balance of the primary producers, but also of the heterotrophic organisms that are in the system, and that we refer to as community production. So it's possible, even if the primary producers are photosynthesizing, your net community production might be zero if the organic matter that's being produced is being eaten at the same rate that it's being produced. So in the remainder of this series of lectures, we're going to talk about these three major factors that influence the amount of primary production in the ocean. We mentioned that the amount of primary production in the ocean is really important to know because of the role of primary production in creating oxygen and the role of primary production in creating organic matter that supports all the food chains of the world, including the food chains that lead to humans. So these are three major factors that affect primary production. In this video, we're only going to talk about light, and turns out that understanding the effects of light on primary production is pretty complicated, so that should be plenty to fill. I want to introduce this term here called attenuation. Attenuation means the diminishment of something. So light attenuation means the suppression of light or the muffling of light. Another type of attenuation that scientists sometimes talk about is the attenuation of sound, how sort of sound fades out as you go a further distance away from the sound source. So water is not perfectly transparent to light. Even if it's pure water, light cannot penetrate an infinite distance between water. It will gradually diminish the more water it passes through. And understanding that rate of attenuation is important for determining how much light is still left at a given depth, because you want to know if there's going to be enough light left at that depth for plants to grow at that depth. And so the obvious thing is that the deeper you go in the ocean, the less light there is, and at some point there's no longer enough light for plants to grow. But exactly what point is that? That's where we need to use a little bit of math. And the math that we use is called the Lambert-Beer equation. And it looks kind of scary, but it's really not that bad as equations go, and I think you'll be able to understand each of the parts of this equation as I explain them. So in this equation, capital E is the letter that stands for light. And we use the word irradiance sometime, and irradiance just means light. Sometimes in this equation they'll use a capital I instead of a capital E, which is exactly the same equation. It's just some people prefer to use an I and some people prefer to use an E. So irradiance, capital E or capital I, is what this means. And it's not just the capital E, it's got a little subscript with it. So here it's a subscript Z, here also you see the subscript Z, or here it's a subscript 0. So the Z is the depth. So if it's 0, it means that's the light level at the surface. If it's Z, it means it's a light level at depth Z. So Z is usually some number in meters. So you could be 1 meter deep or 100 meters deep. And so you're basically figuring out, like, in relation to how deep you are, how much light there is. That's what this whole equation is about. Why do they use the letter Z? Well, it relates to the coordinate plane. So you've probably seen graphs where you've got an x-axis and a y-axis. And if it's a three-dimensional graph, it also has a z-axis. And so if this is the ocean, the x and the y could be sort of like latitude and longitude, and then z is sort of like your depth under the water. There's a little fish. So basically, z equals 0 would be at the surface of the ocean, and the bigger numbers of z would be deeper depths. The other variable in this equation is the lowercase e. It's Euler's number, 2.718. Euler's number is related to natural logarithms, and it's a number that you see in a lot of exponential growth or decay equations. Another variable that's in this equation, and kind of the key variable for describing the light attenuation, is lowercase k, the light attenuation coefficient. So K varies depending on how clear or murky the water is. So if the water is clear, the K is small and light attenuates slowly, meaning that the rate that light fades out as it passes through clear water is not as fast as the rate that light fades out when it passes through murky water, where the K is a large number. So I always like to test out equations to sort of prove to myself that they really work the way I think they do. So I tested out this equation by entering it into an Excel spreadsheet and plotting a graph. This is the graph that I plotted when I put the Lambert-Beer equation into Excel, and I used two different values of K. K equals 0.04 is a typical value for really clear water, like you'd find in the middle of the open ocean gyres, and K equals 0.2 is a value that you might find in murky water near the coast. So you can see that regardless of what the K value is, that light decreases as we go towards deeper depths. But how fast light decreases is dependent on the K value. So with this low K value, you still got a decent amount of light, a little bit left even at 100 meters deep. Whereas with the larger K value, you've pretty much lost all of your light by the time you're just 20 meters deep, which is only about 60 feet deep. If we turn this graph on its side and now put depth on the vertical axis here, this might be a little more intuitive. I also put this color gradient here so you can kind of get the idea that we're talking about sunlit surface waters near z equals 0, and sort of these black lightless waters by the time we're over 100 meters deep. And if you're in these clear open ocean conditions with a low value of K, you still have some light left even when you're pretty deep down. 40 meters deep is probably just a little bit deeper than you could safely go with scuba gear. In the open ocean, there would still be some light at that depth. In murky waters, it would pretty much be black at that depth. So let's put plants back into this picture here. The reason we care about light levels and how they diminish underwater is because we care about whether there's enough light for plants to grow. How much light do plants need to grow? It depends on the plant, but the general rule is that you need about 1% of full-strength sunlight for ocean plants to grow. So by the time you get deep enough that there's less than 1% of the surface light left, there's little or no plant growth occurring anymore. And Understanding this sort of break point where there's no longer enough light to grow requires considering the balance between photosynthesis and respiration, these two terms that we talked about earlier. So photosynthesis being the production of organic matter, respiration being the breakdown of organic matter. So all organisms, including plants, constantly need to be respiring to keep themselves alive. And so they constantly need to be burning organic matter. And it doesn't matter what depth you're at, you need to be burning organic matter. That's why this line indicating respiration is a sort of a constant vertical line as you go through here. But the amount of photosynthesis is dependent on the amount of light. So it's high near the surface, and then it fades out as you go down deeper. There are some complicated reasons why it doesn't just keep increasing as you get into brighter and brighter, and we'll talk about that on the next slide, but don't worry about that right now. Anyways, as you get deeper and the light gets dimmer, you hit some point where photosynthesis is decreasing to the point where it's exactly equal to respiration, and they call that the compensation depth. So your net primary production is 0 at the compensation depth, meaning that your photosynthesis is exactly matching the respiration, and that means that you're not making any more organic matter than you're consuming if you're a plant. And so a plant that was at its compensation point would not be able to grow or reproduce. It would just be able to maintain itself alive. If the plant was a little bit higher up, then it could make gains and it could grow and reproduce. If it was a little bit lower down, it would be running a deficit. So being below the compensation depth doesn't mean you instantly die, but it means that you're not quite meeting your needs on a day-to-day basis, and you're gradually burning up your reserves, and eventually you'll die. Okay, so I hope that explains compensation depth, where sort of the photosynthesis line and the respiration line cross. But there's this other threshold indicated on this graph here, the critical depth. The critical depth is a little bit more complicated, and I'm going to do my best to explain that on the next slides. If you are having trouble understanding the critical depth from my explanation, I encourage you to read in the lecture notes about it, and also look up other resources on it, because the concept of critical depth is not an easy one. Oh, just breaking up the zones here, where photosynthesis exceeds respiration, the plants are able to make gains. Where respiration exceeds photosynthesis, the plants are running a deficit, and at the compensation point. All right, so I'm introducing these concepts on this slide here to get at the idea of what the critical depth is here. But before we can understand the critical depth, we need to know about different zones in the water, and how they relate to that depth. So you can see in the pictures here that there's sort of this light blue water and then dark blue water below the light blue water. So light blue water is, they're calling it the well-lit zone here. A more technical term for it is the euphotic zone. It's a zone where there's enough light for plants to grow. So if plants are in the euphotic zone, they're happy. I mean, plants don't have feelings, but they're able to grow if they're in this sunlit zone. If they're below that, then they don't have enough light to grow, and they're dying or in the process of dying. So those are zones based on light. But there's this other type of zones that we can divide the water up into that are based not on light, but on density of the water. So this zone here above the dotted line is called the mixed layer, and then this zone here where the density begins changing rapidly is called the pycnocline. So if water is all the same density, which it is within this so-called mixed layer zone, that means that the water can mix easily within the mixed layer zone. And so a single-celled organism that was in the mixed layer zone is sort of not just fixed at one elevation, but it's kind of like moving and swirling around at all different depths within the mixed layer zone. And that's sort of what's indicated by the little circular motion here. And In this case, in this diagram here on the left-hand side, the mixed layer zone is pretty much entirely within the well-lit zone. So even as the cell is moving up and down, it's staying within the sunlight and not spending very much time in the darker waters here. So it's able to make net gains. If the mixed layer was thicker though, like in this diagram on the right-hand side, then a cell that was in the water would be spending a lot of its time down in this deep dark water where it's not able to photosynthesize. And so even though when it did happen to be mixing near the surface, it would be photosynthesizing, it would be spending so much of its time running a deficit in this deep dark water that it would not be able to ultimately grow and survive. So whether or not the mixed layer, basically the depth of the mixed layer, whether it's mostly confined within the sunlit zone or extending much deeper into the dark zone, the depth of the mixed layer is very, very important for determining whether a phytoplankton cell is mostly staying in the light or mixing down into the dark a lot. The thing that determines whether the waters can sort of mix or if they're sort of layered off from mixing is this density transition called the pycnocline, which is related to temperature changes or salinity changes in the water. And so it tends to change with seasons as like when the surface waters warm up, that makes them less dense and sort of helps them be separate from the deeper waters and prevents deep mixing. Whereas in the wintertime, there tends to be deeper mixing. So this picture B here is something that you'd be more likely to see in the wintertime when there's not much temperature difference between the surface and deep waters to prevent mixing, and there's also a lot of strong wind that would tend to mix the water as well. So those kind of weather factors can determine how much mixing there is and how deep the mixed layer is, and it turns out that that's very important for whether the phytoplankton live or die. So I'm going to show some pictures on the next page that show the difference in mixed layer depth in different parts of the world over time. This picture here is color-coded by the depth of the mixed layer. So the red and orange and yellow colors indicate a deep mixed layer depth, hundreds of meters deep. So this means that the waters from the surface down to hundreds of meters deep are all swirling together, and any phytoplankton cell that's caught up in that layer is sort of spending a lot of time in deep dark waters. Whereas if the mixed layer depth is shallow, it means that things that are in that mixed layer are mostly within the sunlit zone and have the potential to photosynthesize productively. So this is in January, winter in the northern hemisphere and summer in the southern hemisphere. So you can see the mixed layer depth is deep in the sort of stormy mixed waters of the northern hemisphere, and it's shallow in the southern hemisphere in January. In April, the mixed layer depth begins to get shallower in the northern hemisphere as the winter winds calm down and the surface waters warm up and become more thermally stratified. But the mixed layer depth is deepening in the southern hemisphere as winter comes to those Antarctic waters. And then in June, the northern hemisphere summer, you can see that there's a shallow mixed layer depth in the northern hemisphere and a deep mixed layer depth in the southern hemisphere. And then it begins to change again in November. And of course, this is a cycle that repeats every year. So the idea of the critical depth, which is sometimes also called the critical depth of mixing, is basically the critical depth is the depth that the mixed layer has to be shallower than for a phytoplankton cell in the mixed layer to have net primary production. So if the depth of mixing is like this deep, like it is in this picture, then the phytoplankton cell, which is sort of mixing in the water around these depths, it's mostly in this zone of gains, and its overall net gains are going to be positive. Whereas if that cell is mixing to a deeper depth that's exceeding the mixed layer, exceeding the critical depth, then it's spending a lot of time below the compensation depth and where it's running a deficit, and it's ultimately not going to have positive net primary production. It's going to die. So that's why understanding the depth of mixing in relation to the critical depth is really important for oceanographers trying to predict how productive the primary producer community of the ocean is going to be in that point of the ocean at that time of year. So it's something worth mentioning that the relationship between photosynthesis and light is not always a simple one. So there's some general things that we can predict, like the more light there is, the more photosynthesis there will be, and that's sort of like true up to a point, but because there's a lot of complexities to it, we need to, for every individual type of primary producer, understand in more detail what we call the PE curve, which is the photosynthesis versus irradiance curve. So this is basically like, imagine a graph with photosynthesis on the y-axis and irradiance on the x-axis. So irradiance is the amount of sunlight. So if it's dark, that would be 0 light, and if it's really, really bright, that would be, a lot of light, full strength sunlight. And so obviously if there's no light at all, there's going to be no photosynthesis at all. So there's one point on this graph that we know for sure is going to be 0. And then we would expect that this would go up somewhat as the light increased, but exactly what pattern it would have, we don't know for sure. It might depend on the type of algae. And so I'm going to show you some examples of more realistic photosynthesis versus irradiance curves so you can get an idea of how the pattern of primary production might change for a plant in relation to how much. So here's a more realistic photosynthesis versus irradiance curve, PE curve. So as we said, when there's no light at all, there's no photosynthesis. And when light is very low, there's some photosynthesis, but it's not enough photosynthesis to exceed the amount of respiration. So you have to get to some certain minimum amount of light before you reach that compensation point where now photosynthesis is matching the production of organic matter to the consumption of organic matter by respiration. And then as you get into still more light, you start making gains where now you're photosynthesizing, creating more organic matter than is being used up by respiration. And there's a roughly linear increase in photosynthesis for a while. So just kind of as you probably predicted, more light equals more photosynthesis. What you see though is that after a while it kind of levels off. The light is still getting brighter, but the amount of photosynthesis of this particular algal species, whatever plant that's being represented in this diagram, it's maxed out. It's not getting any higher. And that's because you sort of reach the physiological capacity of that algal cell or plant. It just can't photosynthesize any faster. Its machinery is getting backed up, and so even though there's more light available, it just can't photosynthesize any faster. And if the amount of light keeps going up and up and up, eventually it starts to backfire. The photosynthetic system of the plant gets so overloaded that the rate of photosynthesis actually goes down. The enzymes start running in reverse and things like that, and photosynthesis actually declines. This is something that you might notice if you've ever sort of bought a plant for your house. Usually the plants that you buy for your house are adapted to do well in low light levels because there's usually not a lot of light in your house unless you put the plant right in the window. So we pick house plants to be plants that can survive on low amounts of light. And if you put them in brighter light, it doesn't really help them. In fact, if you put them in direct sunlight like outside, it might hurt them. They might have so much light that they actually start dying. So beware of that if you're ever trying to take care of a houseplant, and because more light is not necessarily always a good thing for some plants. So there's a lot of variation between different species of plants and algae in terms of how they respond to light. So no plant can grow without any light. No photosynthetic plant can grow without any light, but the overall shape of their light response curve does vary between species. So there's some species that are able to pretty efficiently make use of low light levels, and those do well in the shade. But those ones that do well in the shade, they usually sort of top out their photosynthetic rate at a modest level, whereas some of the species that might not do so well in the shade, they can do really well when it gets a little bit brighter because they have a high maximum rate of photosynthesis. So as long as there's enough light, they can just keep photosynthesizing faster and faster. And so if you sort of take these species-specific response curves and sort of overlay them on a gradient of where light be in the ocean, you can see that the species that performs better in brighter lights might dominate waters near the surface, whereas the species that performs better in darker conditions might dominate deeper waters. And we actually do see some of these patterns of changing algal species composition with depth and light, as we would predict from our understanding of their physiology. This may remind you of a niche differentiation graph I showed you in a previous lecture, where I was showing two tree species that were adapted to different levels of temperature, and how one would sort of perform better in hot temperatures and another in cool temperatures, and they sort of partitioned the habitat that way through niche differentiation. Well, it's the same idea here, it's just that the environmental gradient is light now, not temperature. So here we're looking at a couple of broad groups of primary producers in the ocean: blue-green algae, dinoflagellates, green algae, diatoms, and these are really stereotyped light responses for the different groups, but you can get the general idea that some groups like the blue-green bacteria and the dinoflagellates would have an optimum light level at a pretty low light intensity. Other groups like diatoms, their optimum light level would be moderate light intensity. And then some groups like green algae, they're basically the brighter the better for those species, and they would grow well in very shallow waters or even intertidal zones of the ocean. So there's a lot of variation on a finer taxonomic resolution. So like there might be some species of blue-green bacteria that actually would like bright light, but this is just an overall trend of different groups and how they respond to the light. So I've been talking about light as if light is all one thing and there's no difference between one type of light or another, but that's actually not true. So there's all kinds of different light. Light in general is really just a subset of electromagnetic radiation, and it's electromagnetic radiation that has a particular range of wavelengths between 380 and 740 nanometers. So it's kind of hard to visualize, but light functions sort of as a wave, and wavelength is the distance between two humps of the wave. So when we say that visible light is electromagnetic radiation with a wavelength of 380 to 740 nanometers, that means that that distance between, like if we're talking about green light, the distance between the wavelengths of that electromagnetic radiation that we call green light is 500 nanometers. So wavelength is one of the things that we can describe light waves with. And another thing that we can describe light waves with besides the wavelength is the frequency. So frequency means, like, if you're monitoring at a certain point how many waves pass that point, and sort of you're timing it like with a little watch here, the number of waves that pass that set point there in a second is called hertz. So hertz means waves per second. So if you're at 10 to the 0th hertz, 10 to the 0th equals 1. So that means one wave would pass by every second, like wave, 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 wave. That would be 1 hertz. If it's 10 to the 8th hertz, that's like 10 to the 8th waves per second, which is a lot of waves per second. And if you know these two things about the light, the distance between two wave peaks and the number of wave peaks that are passing by a point every second, you can actually figure out the speed of light. So the speed of light is equal to the size of the waves times how many waves are passing by per second. So you just take the wavelength times the hertz, and you can figure out the waves per second. So I matched up the particular frequency that goes with waves of 1 meter wavelength. And so if these waves are 1 meter in size, which is, spread your hands a moderate distance apart, and that's 1 meter, and then 10 to the 8.477 of those 1 meter waves passes by in a second, that means that the speed of light is 10 to the 8.477 meters per second, which is equal to approximately equal to 300 million meters per second. So all different types of electromagnetic radiation actually have the same speed of light. So whether it's radio waves or light waves, they're all going 300 million meters per second, which is really fast. Light takes about 8 minutes to get from the sun to the earth because it's really far, but light waves or any kind of electromagnetic radiation could circle the Earth more than 8 times per second. So it's a long ways around the Earth, and something that's going around the Earth 8 times per second is obviously very fast. 300 million meters per second, very fast. And so that's just a little bit of physics here. So the reason I'm going on this sort of aside about physics and electromagnetic radiation is so you can understand the differences in these different sort of color wavelengths within the light spectrum, because it turns out that the different colors of light have different effects when it comes to photosynthesis due to the way plants harness light. Oh yeah, and you can calculate the speed of light from this chart as 300 million meters per second. Okay, so the main chemical that plants use to capture light energy is called chlorophyll. So chlorophyll, this light capturing chemical, it comes in different forms. There's chlorophyll A, B, C, and D, and each form of chlorophyll captures slightly different wavelengths of light, but in general, the chlorophyll molecules are good at capturing bluish light and reddish light, and not so good at capturing, like green light. So bluish light and reddish light, those are the main wavelengths of light that chlorophyll captures. So all plants have chlorophyll A, that's a basic type of chlorophyll, but some plants have some other types of chlorophyll as well, and the different types of chlorophyll capture slightly different wavelengths of light, which may give those plants an advantage in environments that have a little bit different light spectrum in them. And some plants and algae have extra light-catching chemicals besides just chlorophyll, like xanthophyll, phycocyanin, phycoerythrin, and that helps them capture wavelengths of light that are different than the ones that chlorophyll captures. And that may give them a further advantage in some environments, like where light is really dim or certain wavelengths are filtered out of the water and you need to catch other wavelengths of light. So, most plants that we see on land anyways are green. At least they look green to us. And it's funny because chlorophyll actually does not absorb green light. You could shine just pure green light on a plant and it would die because it's not able to absorb and use that light. And so why is it that a plant, and here's like the leaf of a plant, why is it that it looks green when to our eyes? This is a person's eye, when actually it's not absorbing green wavelengths of light. It's because the light that we see coming off of the plant is what's reflected at us, and so the light that we're seeing bounce off the plant and into our eyeballs is the light that the plant is not absorbing. So the color that something appears to our eyes is actually the color that it's reflecting, not the color that it's absorbing. Okay, so all this wavelengths of light stuff comes into play when we're talking about the light environment underwater. So we know that water attenuates light. Light diminishes as we go deeper in water. But it's not an equal attenuation for all wavelengths of light. So some wavelengths of light are attenuated really rapidly by water. So red light, it is rapidly attenuated by water. So only in very shallow water would you be able to see red things. So by the time you get just a little bit deep underwater, all the red light has been blocked and you can't see red stuff anymore. So if you had like a beautiful red pair of swim trunks and you were snorkeling underwater, you'd see the red color when you were shallow. But if you went a little bit deeper down to where the red wavelengths had all been attenuated away by the water, your swim trunks would just look black or gray. You wouldn't be able to see the red at all. That's one of the reasons why underwater pictures that you take when you're scuba diving aren't that colorful. Everything looks kind of green and blue unless you adjust the colors or bring a flashlight with you underwater to bring out the colors again. Anyways, the wavelengths of light that penetrate furthest into the water are these longer wavelengths of light like green, blue, and violet. So it's nice that plants can absorb blue wavelengths because blue wavelengths of light are the ones that make it furthest underwater. So it's not just water itself that blocks light from penetrating. There are lots of other chemicals that can be dissolved in the water that can change its light blocking properties. And they can be dissolved in the water, or they can be particles that are floating in the water that make the water different colors or murkier. And I'm sure you've noticed water that was more clear or more murky or discolored one way or the other in your observations of the world in your life so far. One of the things that you might have seen, especially if you live in Florida, is that there's some water bodies like in swamps or some estuaries around the mangroves where the water has this really dark and kind of reddish color. It's not necessarily murky, but it has a tint to it, a reddish or amber tint. And that comes from this class of organic molecules called tannins, or colored dissolved organic matter. They're plant chemicals that leach off of decaying sticks and leaves and discolor the water. They look red to us because they reflect red wavelengths, and actually they absorb blue light really well. Because these color dissolved organic matter compounds absorb blue light, they prevent blue light from penetrating very far into the water. And so water that has a lot of this type of dissolved organic material in it may not have a lot of photosynthetic activity because the valuable blue wavelengths for photosynthesis don't make it very far through this kind of water. So if you're a plant and you're trying to grow in these tannic swamp waters, you need to make sure you're really near the surface to be able to get enough light to grow.